When Human Genome Project was launched in late 90s, we had already got some knowledge about DNA and genes. We knew about family of genes. We knew that some genes occur in multiple copies. We knew that genes have spaces in between them. They are promoters, enhancers, non-transcribed sequences. We knew about the fact that in some organisms, the amount of DNA per nucleus is considerably higher than the other. For example, in amphibians, the amount of DNA per nucleus is 40 times greater than that of a human being, whereas a human being is much more complex than an amphibian. So we had considerable knowledge about DNA. We knew about satellite sequences, highly repetitive sequences, moderately repetitive sequences, short sequences, and their position along the chromosome. In the backdrop of all that, we began Human Genome Project. The idea was to find out the sequence of the entire human genome. It took a few years. When that was done, it became known that the human genome has 3 billion base pairs. And 90% of the genome is non-coding. In other words, about only 2% of the genome codes for proteins that we have in our body. Now the question is, what is all that 98% of the DNA doing when it's not coding for anything? Well, this led to speculations, conjectures, surmises. Well, it was thought that maybe some sequences are transcribed for a short period of time. The RNA, the life of RNA is short, half-life is short, they do something or then they're destroyed. Or some sequences are not transcribed at all. But then they reside in the nucleus, the genome, and they are an integral part of the genome. When something is an integral part of the genome, the question arises whether it's really doing something or it's just superfluous, nonsensical, redundant, useless, garbage. Well, it can't be garbage. After all, it has been selected by nature. There must be some kind of purpose behind all this. Now, I'm coming to a different point about variation. If there were no variation, there wouldn't be any biodiversity in the life. And this is what Charles Darwin could not account for very clearly in his theory of natural selection. He didn't quite explain how this variation arises in organisms. But we know that the real source of variation is mutation, and mutation is the change in the base sequence. Change in the base. Suppose there is a word cow, C-O-W. If you replace W with D, cow becomes cod. Cow is a mammal, whereas cod is a fish, meaning it's changed. Similarly, if you bring about a change in the base, and if that base changes the amino acid sequence when the protein biosynthesis is taking place, if the primary sequence of the protein is changed, its three-dimensional structure will be changed, because the three-dimensional structure of the protein depends critically upon the primary structure. And if the three-dimensional structure of the protein is changed, its function will be changed. Now, this protein will perhaps perform differently or will not perform at all. And this will lead to certain changes in the biochemical reactions and finally leading to the change in the character. So this, of course, follows a plan. A sequence. Change somewhere results in the change of the protein, results in the change of the function, and this is mutation. But suppose there is a change in the base sequence of the non-coding sequence. What will have an effect? Perhaps no effect, because it's not transcribing, it's not producing any RNA, and the RNA is not going to be formed into a protein. So there will be no effect on the organism. But would you not call that mutation? Yes, you would. Because after all, mutation by definition means change in the base sequence, change in the base. The big change has indeed occurred, but that change will be called neutral mutation. Similarly, if you change the code, and if the code does not alter the sequence of the amino acid, then of course that mutation will also call neutral mutations. We have large number of neutral mutations, but if mutations, that generally are harmful. If mutations are harmful, of course, then the organism will suffer from it. It depends upon the degree of harm. The harm may be so harsh that it may be lethal to the organism. So the mutation is the source of variation, and the mutation, if it occurs in the coding sequence in the gene, or if that leads to change in the sequence of the amino acid, and if that change brings about change in the three-dimensional structure, which it would do, then of course it will be called real mutation, or the mutation that would bring about real variability.